how we're trying to contribute to better understanding this really unique and in some cases very fragile biological resources that we have. So I'm going to focus, more in this particular talk on some of the cave obligate species. So these are the species that are adapted for living in the complete darkness of caves, right? In many cases, they lack pigment or they lack eyes or have regressed eyes. And in terms of kind of biodiversity in North America in general, I'm specifically focusing in the U.S. and Canada, we have about 1,350 species that have been officially described, and there's at least a few hundred that are probably waiting taxonomic description, and there's many more that we're finding each year that I'm sure that remain to be discovered. So it covers a variety of different groups of organisms. I'm just going to kind of highlight a few of these. Some of the terrestrial species that we have here in the TAG region, for example, just some of the representative groups, and maybe you've seen some of these critters like this. So cave millipedes, we have several different species that are found. Cave pseudoscorpions, many cases, tiny little 1 to 2 millimeter arachnids that in many cases are known from just a single cave system. We have terrestrial cave snails. Again, most of these species are quite small, 1 to 2 millimeters, maybe 3 or 4 max millimeters in size. So if you're moving fast through a cave and not stopping to kind of look around, you're probably not going to see them. We have a variety of different cave beetles. We have springtails. We have bristletails, a lot of spiders, harvestmen, you name it. In terms of aquatic life, maybe you may have seen some of these critters before as well. They're a little more conspicuous because they're a little bit larger. So several different species of cave crayfishes. We have cavefish species, Tennessee cave salamanders in the region, isopods, amphipods, flatworms, a variety of other smaller crustaceans like ostracods and copepods as well that we actually don't know a whole lot about. And so if we look at this diversity just in general, so the triglobions being the terrestrial species, and this is kind of just this is from a review we did a couple of years ago, and already this data is already out to date, is outdated. Alabama, Tennessee, Georgia, just because it's not as many caves, but you kind of see where some of these states rank. So Tennessee being number 1 in terms of the terrestrial species, Alabama number 3, Georgia a little bit further down the list. In terms of aquatic species, Texas is number 1. The Edwards Aquifer, particularly the San Marcos Well region there on Texas State University, is just remarkably diverse. Many crustacean species in particular that are found there. Tennessee, Alabama, Georgia, definitely in the top 10. And then overall, just kind of combining those two groups together, the aquatic and the terrestrial species, Texas is number 1 with almost 250 now. Tennessee's over 200. Alabama kind of ranks 4th, and then Georgia a little bit further down there. But one of the challenges that we face in trying to ultimately conserve and kind of manage these stygobionts and triglobionic species is we don't know actually a whole lot about them. So that might seem maybe a little bit peculiar, but the reality is we know very little about aspects of their ecology, even where they're found. So a case in point, in terms of the Tennessee, there's 11,000 caves, right? I would estimate that we have sufficient biological knowledge for maybe 2% of those cave systems, where we might say we actually have a good understanding of what species are found there. I mean, the vast majority of caves, biologists never entered. In fact, most of maybe some of our observations are made by you all, by the caving community in general. I get emails every week from some of my caving friends who I think I've seen a Tennessee cave salamander in this cave or this new cave, or maybe I've seen a cave crayfish here, or I found this unusual spider or millipede. And obviously, there's a lot of work that we need to do in that front. So one of the other challenges that we have is, and kind of breaks down in this table here, these different kind of biodiversity knowledge shortfalls. So just a quick little overview, just as some examples. The Linnaean shortfall, this is kind of in reference to, we don't have a full characterization of just the true diversity that we have in cave systems. So that includes, we're finding new species right every year. Even in caves that we have visited, biologists visited in the past, we find new species. Similarly, we don't know, we're finding that there's many new species that are morphologically similar to each other. They're known as cryptic species. So they look really, really similar, and that might be expected because of just how harsh the subterranean environment can be for organisms living in there. But it turns out they're quite genetically distinct. Similarly, we don't know much about the distributions of many species. So in some cases, some of these pseudoscorpions that I showed you a picture of, they're only known from a single collection and maybe one or two specimens that are sitting in a jar in a museum. That's it. That's all we know about them. So are they truly just found in that single cave system? Or is it just because we as biologists haven't been able to search, of other area caves? Now, in general, it seems that most cave species have pretty small distributions, but that wouldn't necessarily always be the case. Other aspects of their biology. So, there's a lot of challenges we have in trying to study life underground, but one is trying to estimate how many are there, how many individuals would make up a population. Again, for some of these invertebrates, we just know of just a handful of specimens that were ever collected. So how are they abundant? Do their populations cycle? Are there certain times of the year where you should be trying to survey for them, trying to understand how they're related to each other? What are their physiological preferences? Other aspects of their ecology, what are they eating? What's eating them? All that type of information are virtually lacking for the vast majority of subterranean life, outside of what I refer to as kind of the charismatic cave megafauna. Right, we know more about the cave fishes, the cave crayfishes, the salamanders, for example. And so why do we have these kind of knowledge shortfalls to begin with? Well, part of it is, and you guys I think, are well aware of this, right? Is, trying to access life underground is pretty challenging for the most part. So this is just, you probably even have seen the schematic drawing before, but we can only access, certain parts, within a cave system, right? This is just even looking at the aquatic fauna. We'll be able to sample a spring, a well. We're getting isolated windows into that subterranean realm that's three-dimensional, right? So, for some of these species, they might be utilizing habitats, obviously, that we as humans can't access. So that's part of the challenge. Some other challenges that we face is in terms of subterranean biodiversity as a whole, so including species that are not the cave obligates, we have relatively little funding. And so most of the funding on subterranean biology is maybe unsurprising. It's related to bats and bat caves, particularly protecting bat resources. And of course, they're critically important. But compared to, let's say a small springtail or a pseudoscorpion, we're not sending funds that way for the most part. So that's kind of related to the second point. We do see taxonomic biases in research and also in understanding aspects of kind of the conservation biology of these species. Historically, right, we kind of focus on the larger species, the vertebrates in particular. I mentioned the cave crayfishes, right, things like that, and then the cave obligate taxa. So I say kind of mentioned facetiously here, who cares about cave dwelling copepods or mites, or if you're talking about cave microbiology, there's very little known. We don't know anything about like the protozoans. There's cnidarians, these other groups that can be incredibly prevalent in other ecosystems, have not been studied in caves and other subterranean habitats. In modern times, we kind of have this what's known as a taxonomy crisis. The issue is, for some of these species, they can be really, really challenging to identify morphologically. And we've lost that taxonomic expertise over time. So the taxonomic experts have either retired or they've passed on, and we've not essentially trained and recruited other individuals that have that taxonomic expertise. So we have collections for some groups of things like diplurans and bristletails that are sitting in jars right now that might be 10 or 15 years before someone can actually look at them to be able to say, okay, this is this species, or it's that species, or hey, it's a new undescribed species. Up until really the last 5 years, we've had comparatively few biologists working in caves in North America. So kind of the heyday of North American cave biology was in the 60s, 70s, and 80s. There's been kind of a lull, and this is kind of compared to Europe and Australia and some other areas. That's kind of changed a little bit. There's been several students various programs who are still continuing to work on cave biology in general. Some of the other challenges, right, is many of the caves, as I mentioned, remain to be studied biologically. As I mentioned, just in the kind of tag region, probably less than 3% have been what I would view being comprehensively bio-inventoried. In terms of conservation assessment, threat assessments, the vast majority of these species have not had those assessments conducted yet. And so we see these biases in some of these groups. So this is a figure from a book chapter we had put together, myself and some other colleagues. So these are, I don't expect you to know these. There won't be an exam after this, but these are just some of the major taxonomic groups that we have in caves. So decapods being some of the crayfishes and shrimp, for example. Arachnida includes the mites, the spiders, and pseudoscorpions. hexapods include all the insects, so cave beetles, right? A couple of species of cave fly, and then things like that. So the vast majority there in the left-hand side, the vast majority of the described cave obligate life is arachnids and then the insects, right? Particularly the beetles. But if you look at species that have had conservation assessments, they're on the right-hand side or they're in the middle that are actually listed under the Endangered Species Act, at least here in the United States, you can see that it's predominantly skewed towards amphibians, so salamanders, the Actinopterygi, that is the fishes. They're at the bottom. So those are the vertebrates there. So you see the major bias there, despite accounting for actually a very small percentage of the overall biodiversity of cave obligate life in the U.S. They're disproportionately more likely to have their conservation status assessed and to be potentially listed under the Endangered Species Act. So kind of working in my lab is trying to address some of these knowledge shortfalls. So trying to answer questions like, what are the true levels of biodiversity in different cave and groundwater ecosystems? What sort of factors affect the distributions? Are some of these species actually able to move around from cave to cave? Or are there particular barriers that limit their dispersal? Thinking about aspects of their genetics and how are they related to each other and how have they become adapted to cave systems? And then, finally looking at some of the impacts that we as humans have, not only on the environment, but how that impacts cave life, in general. And then thinking about, are there particular cave systems or cave organisms that we need to be prioritizing because we have very limited, funding and resources available to conserve and protect these species. So, some of the things we've been doing is a number of biological inventories, and a few of you, I think, have been involved with that. I know, I think I saw Mark on the call, the fern cave system, for example. So my lab has been involved with conducting, over almost 750 biosurveys of over 500 cave systems in the TAG region. And again, that's a small percentage, right, of all the known caves that are found here. This is since 2012. We're talking over 20,000 occurrence records of cave life and documenting that's actually well north of 400 species now. It's a little bit older. We have at least 30 new undescribed species that are awaiting taxonomic description. We've sent to different taxonomic experts. At the same time, we've been slowly compiling literature museum records across the eastern U.S. and trying to expand this out for the entirety of North America over time so we can better understand regional patterns and maybe even continental patterns of cave life and cave distributions. And in part, we've been begun developing a cave life database, and ultimately, hopefully, we'll launch here by next summer a web portal that is going to essentially provide an opportunity for cavers and researchers and the general public to query and access some of this data. It's, of course, the cave locations are going to be protected, as well as information on some of the publications that are available for various species or various regions, other educational resources. So as I mentioned, we found some undescribed species, and part of that my lab's been involved with the description of a few species in a way. So the first was related to some of my dissertation work. So I'm actually a Hoosier native. I was born in Bedford, so kind of grew up down there, but then did all my schooling down here in Tennessee. And now, of course, work in Alabama. But during my dissertation work, I did some genetic work sampling populations of the northern cavefish. So it has a unique distribution in that it basically abuts the southern glacial extent of the Pleistocene glacial maxima down to Mammoth Cave region. So a pretty broad range for a fish, and that the range of the species is bisected by the Ohio River. So one of the questions we had was related to whether the Ohio River kind of acts as a barrier for these fishes, or are they somehow able to cross it? What we found was that there was really distinct genetic structures. So we sampled a variety of different populations and were able to kind of show conclusively that the Ohio barrier is a really dramatic barrier for dispersal. You're not seeing fish cross that. And so over the last 600,000 years since the formation of the modern Ohio River, it is essentially cut through that limestone layer, effectively isolating the faunas on both sides. So if you're a white cavefish, yes, you potentially could get washed out in the Ohio. The Ohio is a pretty muddy river, but there's a good chance that you're probably going to get eaten by a bass or a catfish or something like that before you'd ever be able to make it across and be able to make it back underground. Well, subsequently to that, some colleagues and I looked at some existing specimens in terms of the morphology and found there's some differences in body size and body depth and some other features as well, enough that we felt warranted description of the northern populations in the state of Indiana as a new cavefish species. More recently, we found a new aquatic cave snail. Again, this is a thing that's only about 1 to 2 millimeters in size, from just outside Knoxville region. This is in the genus Antrorbus. And so the only other member of this genus is actually found from Manitowoc Cave in DeKalb County, Alabama. So you can kind of see the range there on the left-hand side. So a pretty big distribution gap. And it's likely there's probably other species, or perhaps these same two species somewhere kind of in between. Would not be surprised in the Valley and Ridge, and there might be a cave snail species like this in Georgia somewhere, northwestern Georgia in the Ridge and Valley. Some of the undescribed species that we're still working on, several different cave beetle species, new cave millipede, bristletails, terrestrial isopods, and the list kind of really goes on and on. It's been surprising the number of new taxa that we've found over the last several years. And so that's led to a number of publications, kind of getting some of this information out and available to the scientific community and the general public. So kind of first starting with Kirk Ziegler at the University of the South on some of the Tennessee fauna, looking at patterns of distribution there. We published a couple of years ago a paper on the biodiversity of caves in Georgia, cave life in Georgia. Recent paper in North Carolina. We're working on one for Alabama right now, and then actually doing some work over in the Ozarks as well. So some of the things we're doing with this data set that we're compiling, though, and maybe you've seen some maps like this before as well. The first kind of identifying where are some of the hot spots of biodiversity in terms of cave life, in the U.S. And so maybe unsurprising, here in TAG, right, we're in a hot spot, recognized globally for both terrestrial and aquatic species. So kind of the warmer colors that you see here in these maps, you see TAG really stands out, particularly Marshall, Madison and Jackson County in Alabama, and then Marion, Grundy, and Franklin in Tennessee. You can also probably clearly see the Mammoth Cave region, right? Parts of southern Indiana. There's definitely some biases in these data sets, and they're incomplete. And one of the nice things is we can continually revise these maps as we get more and more data, more and more data in. Some other projects that my students have worked on, recently that not necessarily related to cave obligate species, but in this case, Kayla Wilson, she just graduated, last year. She studied the reproductive ecology of a pretty common salamander that we find in caves. You can definitely find these in Georgia as well. You've probably seen them, the northern slimy salamander. So it's a really common woodland species in eastern North America that frequents caves, but is for a long time we've not really known much about their ecology in caves. One particular population, actually, Cherry Hollow Cave in Marshall County in Alabama, a couple of years ago, we found several different nests of female salamanders. That you see an example there in the upper left-hand corner, female there guarding her eggs. And so Kayla's master's project was essentially tracking these nests essentially for about 1.5 years to see how long were the females kind of staying in the caves, how successful were they at hatching the eggs and rearing the young. So unlike many species of amphibians, slimy salamanders are in a group known as the lungless salamanders. So they actually lack lungs, do all their respiration through their skin. But they also have what's known as direct development. So the eggs are laid on land, not in water. And when they hatch out essentially a miniature adult, as opposed to having like a tadpole stage or a larval stage that would be in the water and then go through metamorphosis. So this is just a video of one of the females that was on a nest. And so some of the other interesting observations, that's actually a cave obligate millipede there that it was eating. So she was able to document some really unique kind of diet observations that we know beforehand very, very little about. So subsequently, I've had another student kind of following up on Kayla's study, trying to understand, well, whether this particular species of salamander cannot actually eat underground in complete darkness. So you may have noticed, right, this is under illumination, that salamander really reacted to that millipede kind of moving around and focused on it. They're really visual predators. So a big question then is kind of like, can they actually feed in the true darkness of caves? And so my other student, Megan Carter, right now, she's been catching salamanders basically putting them under anesthesia, and then stomach flushing them to see what they have in their stomachs when they're caught. And it seems like they actually are not really good predators underground in complete darkness. So these females that might be on their nest for upwards of 5 to 6 months are essentially not feeding or feeding very, very little at all. So they can go a pretty long time without actually eating. Some other kind of discoveries that we've made in the lab. This is I think Mark is actually a co-author on this publication. We discovered a new population of the federally endangered Alabama cave shrimp. So that's actually a photograph of one of the females that we found, essentially in the bottom cave section of Fern Cave. So Fern Cave being the longest cave system right now in Alabama, and also holds the title of the largest graybat hibernacula, winter hibernacula. So it's a really important cave biologically on Wheeler National Wildlife Refuge, specifically the Fern Cave Refuge. So we were kind of surprised to find shrimp there, because it's extends the range into Jackson County, also up in the Paint Rock River Valley, whereas most of the known range is in the Madison County area around Huntsville proper. More recently, we've been doing work at Shelter Cave. So, those you're familiar with Shelter Cave, essentially it's is underneath the old headquarters for the NSS. It was kind of purchased back in 1967 and then gated at that time. It had really diverse aquatic fauna, 3 different species of cave crayfish, Tennessee cave salamander, cave fishes, 2 different species of cave amphipod, the cave shrimp, Alabama cave shrimp as well. And then the populations of most of that life crashed dramatically in the late 60s, early 1970s. One species in particular, the Shelter Cave crayfish, which actually wasn't described until 1997, was one of the smaller of the 3 species of cave crayfish that were found there, and the rarest at the time. Hadn't been seen in about 3 decades. And so many had thought that it might be now extinct. So I'm going to give you a perspective of kind of the decline of the cave life there. So Shelter Cave is interesting if you've never been there. It's essentially a groundwater lake controlled by the water table itself, fed by epikarst, kind of 3 main lake sections. So it's a fair amount of acreage. But John Cooper, when he was doing his dissertation work there, he basically went monthly, if not several times a month, for about 3 years. He would see anywhere from, in terms of total number of crayfish, from 60 to upwards. So I think as high was like 240 crayfish, which is a pretty good number. We are lucky. We've been surveying the cave since 2017, roughly about every couple months. We are lucky if we see 4 crayfish. Usually we see 1 or 2. So we're talking about a substantially dramatic decline. Well, during a couple of those trips, though, we were able to capture really what we could tell was a unusual crayfish. It wasn't the typical southern cave crayfish that we see in a lot of caves in the area. This was much smaller, had a really thin kilo that you see there pictured in B and C. So we captured them, basically took a walking leg as a tissue sample, released them, and so they'll regenerate that when they molt. And we're able to confirm that, based on looking at some of the morphological characters, that this was indeed the Shelter Cave crayfish. So a bit of good news that despite some of the groundwater pollution that's occurred at Shelter Cave and the loss of the grayback colony there, that this species is persisting. And so it kind of gives us hope that perhaps some of the other fauna that used to be found in the Shelter Cave system is still sticking around somewhere. I have a couple other students, Joseph Benito and some other colleagues who have been doing a lot of work with the cave beetles. So Tom Barr, those of you might be familiar, with his work. He was the one who initially published like the caves of Tennessee. But he was a biologist, particularly a coleopterist. He studied beetles in particular and was really interested in the cave beetles, genus Pseudophthalmus and other trichine cave beetles. So he described a number of different species. It had several, but when he passed away, that were in various stages of description, but ultimately never published those manuscripts. So we've been trying to look at some of his work from a molecular perspective, trying to get a better understanding of how these groups diversified. They're particularly diverse in the Richland Valley, as well as the interior low plateau, that you see there. In many cases, you can find 1 or 2 species or more. And to kind of take to the extreme to Kentucky, where within this particular group, you can find upwards of 5 or even 6 species of cave beetle in a single cave system. One of the challenges is they're pretty small and they all look really, really identical. So we've been doing kind of some work on that as well. But what's kind of really taken off in my lab is using some kind of newer approaches to try to study cave life in general. So again, as I mentioned, right, getting underground and trying to access habitats for these, small, 2 millimeter to 5 millimeter beetle or an amphipod can access when you're a 240-pound human like myself can be challenging. So some of you may have heard of environmental DNA or by short, eDNA. So this is this idea, right? As you're sitting in your room right now, you cough, you scratch your head, right? Whatever. You're shedding DNA into the environment. Well, these organisms, as they're living in their environment, are doing the exact same thing at varying degrees. So we potentially can leverage that information by collecting a water sample, basically filtering out using really fine scale filters that DNA kind of sticks to, and then essentially using kind of some genetic and genomic approaches to better understand whether Alabama cave shrimp is found in a particular cave system, right? Or even expanding that and looking at entire cave communities. So environmental DNA is essentially DNA from mitochondria or the nuclei that's essentially released in the environment by an organism. And so it comes in a number of different, factors, different shed skin, hair, feces, gametes, carcasses, you name it. And we can collect it from a number of different sources. So most of this work's been done with water, aquatic samples, and both marine ecosystems, freshwater, groundwater, but we can also collect it from soil sediments, ice cores, biofilms. We've even collected eDNA off swabs of flowers to look at the different pollinating insects, for example, from spider webs. And there's some studies now looking at this, actually just collecting it from the air in caves. So essentially the airflow itself, putting out special filters and seeing what you get. So general idea is we can go to a spring, right? Or we can go, into a cave and sample a stream or a pool, brimstone pool or a drip pool, whatever it might be. We take those water samples, back to the lab and we filter them. Ultimately, we end up with a filter that we extract the DNA from. And then from there, we can kind of take a couple different approaches. So historically, which is, when I say historically, really over the last 10 years, still a relatively modern approach, we focused on single species. So we've been interested in, can we detect this endangered cave crayfish? Can we detect this Tennessee cave salamander? So we would design essentially a DNA assay, amplification assay to target just that species DNA and hopefully amplify just that species DNA and nothing else in that water sample. So we can be able to tell if it's present or absent, and maybe by the abundance of the DNA using quantitative PCR, we can maybe infer something about how abundant they might be. Kind of the other approach, which kind of leverages next generation sequencing technologies, is we can essentially look at the DNA for all organisms that are in that water sample, construct a DNA library, and then sequence all of that using a next generation sequencing approach, and assume it's metabarcoding. So we can get information about not just the Tennessee cave salamander, but also the cave fishes, the crustaceans, perhaps beetles that died in the water, fishes, you name it, from a single water sample. So how can this be used then? Well, we can use it, of course, to detect different species and monitor them, rare species and invasive species, for example. It's being used in a number of different aspects to looking at historic ice cores, so detecting environmental change and how communities have changed over time. Better understanding, if we can look at scat samples, for example, from bats, we can get an understanding of what species of insects or moths, for example, different bat species are eating. And then, from that, obviously connect how this linkages from the surface, to subterranean ecosystems. So there's a number of different potential applications for using this technology. So in terms of kind of groundwater species that this has been applied to, even there's been, again, relatively recently, only dating back to about 2016. The first studies was published on the European cave salamander, aka the Olm, by a couple labs. It's been applied to some Texas Eurecia salamander, groundwater salamanders, a number of different cave fishes that I've been involved with more recently. Several different crayfish species, Alabama cave shrimp, I mentioned, amphipods. So my lab, the ones I kind of highlight here are projects that we've been involved with, and the list is ever growing. So it's becoming more popular, and we've kind of demonstrated now that we can use this approach to detect some of these rare, threatened, endangered species. So one of the first forays with this was with the spring amphipod. So this is the genus Stegobromys, a really, really diverse group of amphipods. They all look really, really similar. So you might be wondering, like, what the hell's going on in that picture there? So in the DC area where this particular species live, around Rock Creek, the actual type locality is pretty close to the National Zoo, if you're familiar with that area at all. They live in these Hepatomenorrheic springs. So this is essentially, or seeps that you see right there. So that's Dave Culver and Tanya Pepon. And essentially it's a wet spot in the woods, right? So water is kind of percolating back on the surface. And so the historic way to try to study the species was to basically grab the leaf litter, pick through it, find some amphipods, take them back to the lab, and then look at them under a microscope to see what species you have. So one of the challenges, though, is that this rare endangered species co-occurs in these habitats with like 4 other amphipod species. So Fish and Wildlife Service would not allow them to collect more than 10 amphipods at a time. So they could collect 10, and if all 10 happen to be one of the other species, they could go back and collect 10 more. Well, if they find 2 in the next round, they could then collect 8. And then if they found 2 more, they could only collect 6. So you get the idea. So it's a pretty laborious process. So eDNA potentially could be a kind of an attractive way to help save money and more efficiently kind of survey these type of habitats. And so we were actually able to demonstrate that at existing sites where they were known from, that we could detect their DNA, from these seep water samples. And we also found a couple new sites, not necessarily significantly expanding the range, but showing how this technology could be used. More recently, my graduate students and undergraduate students have been involved with developing some assays for some of the cave crayfish species here in northern Alabama. We have several species that are of conservation concern, one being the Sweet Home Alabama cave crayfish, Cambarus cupi. So we were able to detect its eDNA at a couple of different sites, historic sites as well as a couple of new sites. Again, not necessarily significantly expanding its range. So Spencer Boyd's master's project was on this. More recently, Katie Dooley kind of expanded that project, sampled several more cave systems, and also developed an assay for the Alabama cave crayfish, another endangered cave crayfish in the state. And made a kind of really surprising discovery here. So the Alabama cave crayfish, although being again a species of conservation concern, had a fairly broad range, essentially along the Tennessee River, from about Huntsville over to Florence, thereabouts. Whereas Sweet Home Alabama cave crayfish is only known from 4 sites just in Marshall County. So she developed for each of our each water sample that we were processing, each spring or cave system that we sampled, she ran an assay for both species. And we were expecting, some of these sites around Huntsville to be Cambarus jonesii, and we weren't getting any positive hits. Instead, we were getting positive results for the Sweet Home Alabama cave crayfish, so Cambarus cupi, which is a little bit surprising. So they look really, really similar, and actually appears to be kind of a species complex. We went back to some of these sites and actually got some tissue samples from crayfish, and were able to show that basically the field guide ranges for these 2 species are completely wrong. Turns out that the Alabama cave crayfish has a much smaller range now distribution, whereas the Sweet Home Alabama cave crayfish has a much larger distribution. So we were able to kind of use environmental DNA to help us better understand the ranges of these species. I mentioned Alabama cave shrimp. We conducted similar type of study there, sampling a number of different sites, and unfortunately didn't find any new sites outside of Haley Spring Cave, which is hydrologically connected to the Fern Cave system. But we again hold out hope that maybe we'll find some new sites for this fairly endangered species. Similarly, we designed an assay for the Alabama cave shrimp, which is only known from Key Cave there outside Florence in Lauderdale County, Alabama. We were able to detect it at its type locality where it's found, but nowhere else. So what are we wanting to kind of do, kind of from here, with this kind of technology, these approaches? So a big question is trying to understand basically how long does environmental DNA kind of persist in these environments? So on the surface, as you're shedding your DNA and it's hot out, it probably breaks down in the order of hours to days to maybe a couple weeks, kind of depending on things like temperature, UV light exposure, microbial activity, things like that. In the darkness of caves, we kind of lack UV light. Temperatures are pretty stable. We generally have lower microbial activity that's going to be digesting that DNA, breaking it up. So it might potentially persist for a much longer period of time, which might allow us to leverage more genetic information from the environment than we say couldn't do in a surface habitat, for example. So we have a project going on at Shelter Cave where we've set up a series of mesocosms. We've established they're basically 10-gallon buckets, from Lowe's. We filled them with cave water there, and we're kind of simulating what would happen if a cave crayfish in like a rimstone pool died. How long will we be able to detect its DNA, over time? So I ruined my wife's blender by buying some lobster tails at Kroger, blended them together, and basically inoculated each of these buckets. Essentially, one of these sets of buckets at the entrance, one at the edge of the twilight zone, and then one in the dark zone itself. And so we've been following this. This is just showing some older data now for quite a bit of time. And so we were taking initially weekly measurements, and then using quantitative PCR, we designed an assay, right, to detect the eDNA. And so this kind of summarizes what we kind of see here. At the red is the entrance array, green the twilight, blue the deep zone, kind of over time, looking at eDNA concentration. And so there's some variation there. So one of the challenges we had during a really wet, rainy period is some of the drip water was got in our buckets and like diluted our water or diluted our mesocosms a bit. So we kind of see that dip that then recovered. But in general, you kind of see a slow decay there. But we're talking about almost a year, right? Almost 270 days since we started the project, and we're still detecting its DNA. Whereas, again, there's been a number of studies looked at this in surface ecosystems for a variety of different organisms. We're talking, days to weeks, maybe. Well, I can tell you that we've been doing this project now for over 3 years. We're still detecting environmental DNA from this lobster. And so what this potentially suggests is that certain cave systems might act as kind of a DNA museum, so to speak. It can last for a really long period of time. And that has obviously implications if you're trying to develop one of these assays, right, to study a rare, threatened, or endangered species. We kind of want to know if we detect its DNA, that means that species was there the day that we sampled, right, or was there at least yesterday, not necessarily 3+ years ago, because we might be actually missing when population might be declining or something like that. One of the other things that my lab's slowly starting to work on now is to rather than kind of doing these single species assays, right, trying to look at entire cave communities. So developing assays where we can understand or get some information about the biodiversity across many, many different groups of organisms, not just, Tennessee cave salamander, but also all the crustaceans, all the annelid worms, right? Potentially other invertebrates, as an example. Now, one of the challenges that we have with that is that this approach, when we generate this DNA data, is we kind of need to have some sort of reference database of genetic sequences that we can compare to. That way we can match up this unknown sequence that we got in the sample, and we can say with some confidence, oh, it's 98% similar to a Tennessee cave salamander, so it's probably a Tennessee cave salamander, right? One of the challenges, though, is for many different groups of particularly invertebrates, from the isopods to the ostracods to the flatworms and you name it, we don't have that reference sequence database. So this is something that my lab's slowly trying to develop, and we're trying to get some other labs to kind of assist with this. One of the other challenges we have too is I mentioned kind of the cryptic diversity, right? So for some of these species where we're more, species that we recognize morphologically, or there might be genetically really divergent and distinct lineages within them. So that can be another complication that we're trying to address. So kind of related to that is part of my National Science Foundation grant. We kind of have an outreach program. We're developing some kits that we're going to send out, essentially kits to local school systems within the tag region where they can go sample a spring, or if they do have access to a cave, say one of the students, might have a cave on their property, they can collect a water sample. They can filter it on site and then send that back to us, where we'll essentially incorporate it into our pipeline. And then share that information, that data, with the classes as well, with the teachers and the students. And then finally, I mentioned that cave about data portal. This is something that I'm hoping long-term will be a valuable resource, not just for researchers, but also for cavers who are interested in biology and the general public. So it's kind of a central place where you can look at information about individual species or find different publications or reports. And access to genetic data and a number of things there. So we're compiling species occurrence data from a variety of different sources, from the literature, as well as some of these other initiatives that have been developed for just biodiversity, in general. And obviously, one of the complications and challenges that we have with cave data, right, is it's often quite sensitive and shouldn't be in the hands of the general public, and to varying degrees. So we're taking precautions with that of who can have access to certain types of data, right? So for my own research, right, I have specific data use agreements with the Tennessee Cave Survey, Alabama Cave Survey, and whatnot to use it for my own purposes, for my own research. So, there's those types of things we need to ultimately, work out. But again, I'm hoping this is going to be a really valuable resource, in the future. So with that, just like to thank a lot of members of my lab who've been involved with predominantly cave biology projects. I have some students that work on other things like snake communities at Wheeler National Wildlife Refuge or box turtles and things of that nature. My work wouldn't be possible though with a lot of collaborations with not just other wonderful colleagues and scientists, but this is just this short list, a number of cavers who have been instrumental in helping me with my research, and many that end up being co-authors of publications. And I can't thank them. I can't thank them enough. So with that, I'll be happy to answer any questions y'all might have, and I really appreciate the opportunity to talk to y'all.
Matt, what an awesome presentation. Thank you so much. Cavers, question and answer time.
Okay.
And if I can't hear you, you can type in the chat and I can try to answer it that way too, if we're having any connection issues.
Zoom can be finicky with cellular data plans. That can be a real inconvenience. So
you would think by like 3 years into the pandemic that we'd all be experts with Zoom and other technologies like this, but I found that is absolutely not the case.
Well, Matthew, I have a couple of questions.
Sure.
So I know for above ground life, geography plays a major role in like splitting of species. How does that compare to subterranean life? I know you said like the Ohio River kind of caused a divergence of species there. Do we see that more so or less so underground?
So it kind of depends on some of the different taxonomic groups, right? In some cases, we see, like if we're looking at surface fishes versus cave fishes, some of those hydrological divides shape kind of both groups. But in other cases, particularly like out in the Ozarks in Missouri, you see a lot of pirating, right? Some of the adjacent surface drainages that we can gain some insights into that by looking at some of the genetic structure of some of these organisms. So when we don't see necessarily an apparent pattern that might match what surface species might have and stream drainages, that might suggest that there's greater connectivity in some of these areas. In other cases, it's more the opposite, right? Some of these species appear to be really restricting their ability to even move from one cave system to another within the same watershed or even in the same ridge line. Some of the aquatic species. Now, in other cases, I mentioned some of the pseudoscorpions. So many of them are endemic to a single cave. One that is kind of the exception to this is the southeastern cave pseudoscorpion. So it's one that's often found around bat guano, wood rat nests, things like that. You guys, if you've ever played around with any of that, you may have seen them before. It's a single species right now that's known from essentially Indiana down to Georgia and Alabama, right? Pretty broad range. So the thought was, a graduate student, Charles Stevens, he just graduated now. He's now working over in Missouri. His dissertation work was looking at that species in particular. And so it thought was we'd probably find these cryptic species, right? You'd probably find several different species. Maybe it's associated with hydrological boundaries, whatever it might be. It turns out that it's all the same thing. So one of the big questions is like, how is that possible? How are these things moving from a cave in Alabama and ultimately be able to spread all the way to southern Indiana? And we don't really have a good answer. So for other pseudoscorpions, they're known as it's known as foracy. They'll actually basically grab a hold of another insect, particularly flying insect, and then hitchhike somewhere else. We've never been able to find the species outside a cave, though. It's never been collected in the leaf litter anywhere else. So it's kind of a puzzle right now. Are they using bats? Are they using raccoons? Is it insects, flies? We don't really know the mechanism. They're not crawling. We're pretty sure, right? Because they're pretty. They're not real fast, right? So they're kind of like a snail. They're not getting around real well in that regard. So yeah, it's real variable depending on some of the species.
Yeah, that's interesting. Thank you.
So I think Thomas had a question in the chat, is asking, what should cavers think about when caving to preserve the ecosystem in caves? Should we avoid stepping in pools, etc.? It's like anywhere else, right? Anywhere where we're potentially stepping, there potentially could be disturbing, habitat. But the reality is, if we stick to certain areas, high traffic areas, and just stick to that for the most part, I think we're in good shape. Some of the issues and some of the more challenging issues are in some aquatic stream caves where I would. How many of you have been to Bluff River in Alabama? Are familiar with the cave? So pretty popular cave, easy to get to, real pretty, a lot of bio-life, right? But you have to kind of like traverse, through the cave stream for the most part. Well, yeah, when you're stepping on rocks and things like that, there's the risk of trampling on or smashing, anything that lives underneath. And so, tread lightly is my best advice. I mean, we can't protect everything outside of like closing off caves, and that's kind of defeats the purpose. We wouldn't know about life in caves if they weren't open. And I'm very much anti closing caves, to the extent that respectful cavers like you all are not the problem. It's caves where you get these spelunkers, right? The amateurs spelunkers who, you guys have experienced this too, who are wearing flip flops and have shorts on and a handheld flashlight that you encounter, almost a mile back in, Limrock, which I've had happen. It's just ridiculous. It's those folks who usually are the more problematic individuals. And so, I definitely don't like to put gates on caves, but sometimes it is required to keep out folks like that. Or if there are really, really sensitive species. So of course, bats in the wintertime, they can be really, really sensitive in terms of disturbance like that. We honestly don't know about how our presence might impact some of these other invertebrate species. We presume not a whole lot, but we honestly don't know. And I can't give an answer to that. Any other questions?
Going once.
I was also curious about the fossil record, and how much you're able to use that for cave life.
So for cave obligate species, virtually zero. That for a lot of these invertebrates, right? They don't preserve well. You're not going to find a fossilized amphipod, right? Cave amphipod anyway. That doesn't mean that we can't make some inferences about that by looking at related surface species and if there's a fossil record for some of those groups. Maybe some of the exceptions might be, some of the cave fishes and the salamanders. There's a bit more, but a more robust fossil record, not for the cave species, but at least for relatives. But we face these challenges for even like the fossil record for just surface beetles is really, really poor, right? Some groups just don't fossilize well at all. So it's, in many cases, we don't know when particular groups of organisms went underground. Was it 10,000 years ago? Had they been underground for 5 million years? Is it longer than that? We can try to make inferences, based on how much divergence there is with other species, but that only tells us when they kind of were isolated from each other, and is that associated with when they went underground or not? That's to be determined, right? So it often requires information about the geology, relying on, karst hydrologists and others and other geologists to put estimates on how old cave passages are, for example. So we really have to work with other individuals like that who have that expertise to try to put the whole puzzle together. And then sometimes we get new information and have to completely change it. So for the longest time, right, it was kind of thought that a lot of temperate cave faunas were influenced by the Pleistocene glaciation. So maybe they're relatively young, maybe no more than a couple million years old. And it seems like, at least for many of these groups, we've looked at some of the genetic data, they date back well before the Pleistocene. So a lot of the cave fauna may have been established before then. Doesn't mean that the Pleistocene glaciation didn't have an influence, but some of these major groups were already kind of in place. Now, exactly when, was it 5 million years ago, 10 million years ago? We're still trying to figure that out.
Awesome. Yeah, thank you.
Other questions?
Well, if there's no other questions, yeah, thanks again, and hope to get underground with y'all if you guys are ever interested. And I've been underground with Mark at least a couple times, and would love to have you along. It's always great to have folks like yourself who have an interest in biology and want to learn more, because I learned a lot from you all too.
Well, thank you.
Thank you so much.
Yeah, thank you, Matt. That was great.
Yeah, thanks, guys and gals. Guess I'll talk to you later.
Thanks again, Dr. Niemiller.
Take care.
Bye. Wow. I love getting experts in on these meetings. It's incredible. Okay, so moving right along. We've already done the new member introductions, I think. I'll say, don't forget to join the NSS. You can be a full member of DCG, and that comes with perks. Beyond that, since we do have a new caver in this meeting tonight, I'm going to do my thing and read the caver's code real quick. It starts, can you keep a secret? We pride ourselves on being on the down low. Cavers don't broadcast these special places on social media in order to protect them. Caves are special. If you want to know more about them, you'll find us. And look, you did. Leave no trace, none. No tobacco in caves, no trash, no poop or pee. Leave only careful footprints, and that means keeping your trip small and being willing to do something else if the cave you're at is crowded. Most southeastern caves are on someone's private land. Many are closed. More will get closed if we aren't on our best behavior. Always get permission and don't trespass. Don't yell. Don't cuss too loud. Don't get naked where you can be seen. Park out of the way and don't block roads or gates. Cavers do right, and it's important that we live up to that. And beyond that, let new cavers know we do have a rental helmet program for those of you who don't have much equipment so far. And the way that works is there are 2 sets of rental equipment. We have both children's and adults caving helmets, and they come with headlamps. I keep a set with me, and Nancy and Brent keep a set with them. And the way it works is it's $5 per helmet per weekend. I believe that puts us right about the end of the program. Officers, is there anything else that we need to go over? Going once? Going twice? Okay. I'll leave her at the end. Chair motions to adjourn the meeting.
Second.
Okay, we got a second. Waiting for objections. Going once. Going twice. All right, everybody. We are adjourned. Thanks for joining, and have a great evening. And we look forward to seeing you underground.
Thanks, Joe.